So the last time that we got together, we looked at this idea of the energy intake. And remember that energy intake comes from the food that we eat, specifically from carbohydrate and from fat. So the carbohydrate and fat is taken into our bodies. Our bodies can digest that food, put it into our bloodstreams and deliver it to the cell. So that's the energy intake. Then we need to think about energy used. And remember that all of our cells have got a whole raft of chemical reactions, which are called the reactions of metabolism. They allow us to do Mrs. H. Gren, and all the reactions of our body require energy. So our basal metabolic rate is the kind of the standard benchmark energy that we need. And then if you're more active and you do lots of running around, you need even more energy to be intake because you're using up more energy. So the idea is that the energy that you take in for food should be equal to and balanced with the energy that's used by the body. That's going to keep you healthy, and it's going to give you your weight's not going to fluctuate. If the energy that you intake is greater than the energy that your body cells use, then you are going to put on weight, and there are all sorts of health implications for people who are obese. And also, if the energy that you take in is too little to meet the energy that is required by your body, then you're going to lose weight, and that also has implications for health. So we want to have the situation where the energy that we take in via our food is appropriate for us as individuals in terms of the energy that we use up. And if we jump down to this number 3 here, different people have different energy needs. And remember that last time we used the example of the pregnant lady. So if we compared when she's pregnant to when she's not pregnant, her energy requirements when she's pregnant are going to be greater because she needs energy to meet her own metabolic functions. She also needs extra energy to give to the baby so that the baby can develop and grow appropriately. We talked about the way in which our cells can use energy for our metabolic processes. And we talked last time about this chemical reaction of aerobic respiration. So cells in your body are doing aerobic respiration, and they do that by taking in glucose. They react it with oxygen, and they do a chemical process which allows them to make ATP. And ATP is kind of the energy currency of the cell. ATP allows the chemical reactions, the other chemical reactions in the body to take place. It allows you to do muscle contraction. It allows you to grow, etc., etc. And these two, carbon dioxide, are waste which we breathe out. So respiration, remember, is a chemical process that takes place in cells to enable us to produce usable energy. Aerobic respiration is all about transforming the chemical energy that we've got in food into this form of energy called ATP that can allow our bodies to do the work that we need our bodies to do to stay healthy. We also introduced last time this chemical reaction, and this is the syllabus statement that we're going to be focusing on both today and also next lesson as well. Now, this capital B here is signifying that this is a triple science part of the course. Is quite difficult, therefore. And this experiment we're all going to do, the experiment is outlined in page 13 of your textbook. And I'm going to show you in a minute a video that will allow you, again, to think about this experiment in context. So we talked last time about this idea that burning food or combustion of food and heat energy being produced was a model for respiration. So in respiration, we take food, we combine it with oxygen, and we make ATP. If we're doing this process, we're taking food and all of the chemical potential energy which is in food, and we are burning it with oxygen, very similar to respiration, but all the energy produced is in the form of heat. But we can absorb the heat into this water, we can measure that heat change, we can plug everything into this chemical reaction here, and we can get an idea of the energy content of food. So what we're going to be doing today and next time is unpacking this particular investigation. But before we get stuck in, I want us to think about what we did in year 7 and year 8 about scientific variables. And some of you might have got a good grasp of scientific variables, others might still be a little bit uncertain. This is a video, a YouTube clip. It's posted in our Google Classroom for you to open up and you can play it. But what I want you to think about is to pause the video in just a moment and think, do I know what an independent variable is? Do I know what a dependent variable is? Am I clear about what is meant by a control variable? So the scientific variables are the independent variable, the dependent variable, and the control variables. Now, if you can't define those, you need to stop this video now, jump into the Google Classroom, remind yourself by playing this video, email me, ask any questions in Google Meet to make sure that before we go any further, this idea of scientific variables, you've got it nailed down. So these are definitions that might be useful for you to add to your booklet. But the independent variable is the variable that you are deliberately, purposely changing. It's the variable that you want to find out about. It represents the cause or the reason for a particular outcome. So in this experiment, if you did watch that video, it was the volume of water. So he was making sure that the volume of water was the only thing that was changed. The volume of water in that video clip was the independent variable. And when we plot independent variables, we always plot them on the x-axis, the horizontal axis of a graph. The dependent variable in that video clip was the growth of the plant. So he's deliberately changing the amount of water that's given, and the outcome, or the dependent variable, is the growth of the plant. So the variable that responds to the change, the independent variable, the thing that changes in response to the volume of water, is the dependent variable, and this is always graphed on the y-axis. The control variables are all of the factors which you keep the same during the experiment. So the only thing that you're going to change deliberately is the independent variable. Everything else needs to be kept the same. So in this particular video clip, he kept the quantity of light that the plant had the same. He kept the conditions that the plant was in the same, the temperature of the room, etc. So if you keep all of the control variables standard across all the experiments, you only change this, what you've got is a fair test. So these are good definitions and one that I suggest that you write down. So a fair test is one where you change one variable, which is the independent variable, and you control all the other variables. So again, just to review what happened in that video clip, he had different plants. The independent variable was the volume of water. So each one of these was given a different volume of water. The outcome, the dependent variable, was the growth of the plant, which was measured. The control variables were all kept the same. So the only thing that was changed was the volume of water. Therefore, this was a fair test. Now, the next part is again thinking about experimental design, and there are three terms that are here that you need to be clear in your minds about. So again, I'm going to ask you to pause the video, and I'm going to ask you to think whether you know what precision, accuracy, and reliability means in terms of experimental design. So I would suggest you pause the video, you write it down. If you're not sure about these, then come back into this video and I'm going to unpack it in a second. And again, post some questions in the Google Classroom if you need extra support. Come into the Google Meet and we can discuss this further. So precision. If I want to look at the example of taking a measurement of temperature, I could use this particular thermometer here, and temperature has got a unit of degrees centigrade or degrees Fahrenheit, and for us in science we like to use degrees centigrade. That's the standard unit that we use in science. And what we can see here is that this is going to increase, this blue liquid is going to increase up here as the temperature increases, and it gives us an idea of value of temperature in degrees centigrade. Now if you look here, this is 0, and then we go up to 10 to 20, and we've got the different units of measurement just here. So hopefully you can see that there are 5 small divisions between 0 and 10, which means that each division is worth 2 degrees centigrade. So we could take the temperature, for example, of water using a thermometer, using this machine, and this is only going to be accurate really to 2 degrees centigrade. Now by comparison, we've got this piece of equipment here. This is a digital thermometer. This is the kind of thing that you might see in Hong Kong at the moment where people are checking people's temperature as an indication of infection. And what you can see is this digital thermometer is reading to 1 decimal place. So we say that this piece of equipment is more precise than this piece of equipment. So in this particular example, this has got a higher degree of precision. So here it tells you what precision is. It's really all about the instrument, and it's all about the number of decimal places that can be given for a particular instrument. So if you want a more precise measurement, you would use a digital thermometer over a regular classroom thermometer, which looks more like this. The next term is accuracy. Now, what we got here is this idea, again, of temperature. But if you're going to bath a baby, you want the temperature of the water not to be hot because you don't want to boil the baby, but you don't want to freeze the baby either because babies are very, very sensitive to temperature. So the temperature of the water is going to be just right. So what this lady is doing, she's popping her elbow in to take an assessment of the temperature of the water. By comparison, what's going on here, you can actually buy a baby bath thermometer. So this little duck here floats on the water and it gives you a reading a little bit like the last time to 1 decimal place. Now, the word accuracy is similar but not quite the same as precision. So the accuracy is how close your experimental result is to the true value. Well, obviously, if the precision of the instrument is greater, then your result is going to be more accurate. So let's say that again, just to be clear. Accuracy describes the difference in the measurements that you are taking scientifically and the actual true value. Precision is measurement itself and the equipment that is used for the measurement. So the best scientific observations are both accurate and they are precise. Now, to check on our precision, what we tend to do are repeat tests. And repeated tests give us an idea of reliability. So the reliability is how similar the results are if you repeated them with the same methodology. So what we've got here, don't worry too much about the actual experiment, but here we've got a Petri dish which has got agar, and we've got little discs here, and these discs are containing mouthwash. So we're changing, the independent variable is the concentration of mouthwash as a percentage. So the independent variable is here, and the independent variable is always given in the first column of the table, by the way. Then what we've got is the dependent variable over here. So the diameter of the clear area around the mouthwash is taken in a unit of millimeters, and the larger the diameter, which is clear, so you can see this one is a larger diameter compared to this one, that gives you an indication that this one is more effective. So what we're doing here is we've got test 1, 2, and 3. So we've got the independent variable, and then we've got 3 trials in this particular case for the dependent variable. And in each case, we're measuring it with a ruler, and we're looking at the millimeter. And if we look at the concentration here of the mouthwash 0, we can see that the repeats are exactly the same. They are very, very consistent. Then what we've got at 20 degrees is there's a little bit of variability here, but it's not too broad. Then we've got, if we skip down to 60, we've got 10, 2, and 12. Now, this is clearly a problem. This is clearly what we're going to refer to as a rogue result or an anomalous result. And what we do with an anomalous result is we ignore it. We say that this one and this one are close enough, they are consistent enough. This one is too far away from the other patterns that we've got, so we're going to ignore this. So the calculation of the average or the calculation of the mean always excludes the anomaly. So the reliability of something, the takeaways are here. If results are similar each time, which they are exactly here, we can say that the results are consistent. And if the results are consistent, then the results are reliable. The repeat tests allow you to spot rogue results, anomalies, odd ones out. We exclude those anomalies in terms of calculation of the average. So hopefully those terms now are a little bit clearer in your mind. So we know about variables, and we know about precision, accuracy, and reliability. The next thing that I want you to do is in the Google Classroom, you've got this document, it's a Word document. You need to download it and you need to read it, and we'll meet again. This one also we talked about last time. This is in your textbook. I would suggest that you read this one too until we meet next time. And finally, this one here, this is again a video that's posted into the Google Classroom, and you need to watch this. This is actually this syllabus statement unpacked. So this experiment here is being done within that video. So watch that video and think about